Thanks very much, Emily, and thank you for the invitation to be here. It's lovely to come and speak to you all. I was reflecting during the opening of this session that I first started talking to Emily about my ideas 25 years ago as a PhD student at the Institute of Child Health, working with Jim Stevenson, doing a twin study of anxiety and depression, and she initiated the Genetics Special Interest Group, and we used to meet over on Windsor Walk once a month and talk about genetic analyses, which at that point for psychiatry really meant twin analyses. We didn't really have any other methods, but things are quite different now. So it's interesting to reflect on that. So yes, my talk is about therapygenetics, and I thought I'd start off by telling you what that is, what the word means. You may well have not heard it before. So when we think about individuals with anxiety and depression, which are the disorders I work with, a lot of them are treated with psychological treatments such as CBT, and we know that these are effective treatments, but they're not 100% effective. We find that roughly half of the people treated with CBT through the NHS will recover from their conditions sufficiently to return to education or return to work if they're adults. But that leaves the 50% who have not made sufficient recovery to be able to engage in their daily activities. These individuals continue to experience a high degree of impairment alongside their symptoms. So it's a really huge research area at the moment to try and predict who is not responding to these treatments. It's the same question that Bruce started his talk with. And so one really major goal of my research team is to try and understand what individual factors predict poor outcome to psychological treatment, particularly cognitive behaviour therapy. And by adding genetics to the predictors, we look at a whole different level of risk, really. So it's traditional to look at clinical factors, it's traditional to look at demographic factors such as age, but it's not very usual when thinking about predictors of psychological treatment outcome to think about genetics. So that's what therapy genetics is, predicting therapy outcomes from genetic factors alongside other factors. We certainly don't look at them in isolation. And so just to say a few brief words about the scale of anxiety and depression, they are two of the most common mental health problems, and between them they affect around a quarter to a third of the population at some point during their lives. So these are a huge, huge burden, both to the families they affect and to society as a whole. They start very young. Around half of anxiety disorders start before the age of 11. So these are affecting our children in our primary schools. That's very young, and a lot of people not in child and adolescent mental health are very surprised by that. This audience perhaps less so, but just to remind you of the importance of tackling these problems very early. And depression, around half of the cases have occurred by 19 years or so, 21 years or so. So by the time young people are entering adult life, and particularly by the time they're leaving the opportunity to be referred to CAMHS, over half of them will already have experienced, perhaps even 3/4 will already have experienced their first episode. So there's a real importance for us as a group to be serving these young people as well as we can and trying to help them emerge into adult life as young adults with good mental health. Because these difficulties show huge stability into adult life where they have major knock-on effects, particularly on employment capacity. And this has led to a lot of interest. The huge prevalence and impairment associated with these disorders has led to a lot of interest in the cost of them to the society. So these are figures for the UK. The cost to the UK economy. This is a prediction to 2027 based on current data, and in the blue bar at the bottom of each of these we've got the direct costs of treatment for depression, anxiety, schizophrenia and bipolar. But in the green part of each bar are the indirect costs. Now these are of course much harder to estimate, but they cover things such as loss of earnings and uptake of benefits, and you can see they're absolutely huge for depression and anxiety, 4 or 5 times the size that they are for schizophrenia and bipolar, and that's partly because they're so common. And so this is a really major problem, and it has led to a real sea change in how the government sees these disorders, because of course they are quite often motivated by things that have a financial impact. So this led to the development of the Improving Access to Psychological Treatment service in the UK, which started off initially for adults And within the adult service, there are two intensities of treatment. Low intensity, which in the case of children often applies to group treatments, where you might see a group of 4, 5, 6 children together. This is very common for treatment of child anxiety with CBT. And other low intensity approaches are, for example, to have the mum work through a resource of some kind, a book or an online resource with some more remote therapist assistance. And obviously the cost of these sorts of services to both child patients and adult patient services is much lower than it is for high intensity, which is your traditional one-to-one with an individual therapist, which is clearly a far more expensive approach to treatment. And so one of the major questions facing the NHS at the moment is to try and identify within the IAP service which individuals need high intensity treatment and which need low intensity. There's not necessarily quite that same clear-cut distinction in the child service, but I've given you the illustration for the child service because the data I'm going to show you relates to children, and I think the issues apply there equally well. So who does need high-intensity treatment? Who warrants having that greater expense put into their mental health? Well, the primary variable that is used to make this decision, at least in adult services at present, is severity. So most individuals going into the IAP service have a stepped care model. They start off with low intensity and move to high intensity. But the more severe end of the distribution are, where possible, immediately given access to high intensity. We don't actually know if that's necessarily the right approach, but we do know that high severity is associated with poorer outcomes, so it does seem reasonable. Another thing associated with poorer outcomes is comorbidity. So anxiety disorders in the presence of a comorbid depressive disorder are the hardest group to treat out of those with anxiety and depression. But might genetics also be a way of predicting, might that also be a variable associated with poor treatment response alongside severity and depression? And the reason we think it might be is that we know that both severity and comorbidity themselves are influenced by genetic risk. So individual patients with more severe symptoms or more stable, more chronic symptoms, individual patients with a higher degree of burden, more comorbid disorders, show a greater level of genetic risk. And so it seems reasonable that if it's these more severe patients that are having poorer outcomes from services, then tapping into the underlying genetic risks may be a useful way to contribute to our understanding of outcome. So this was the rationale behind a study that I set up that involved collaborations across 12 different child anxiety clinics internationally. You'll recognise many of the names of the group leads. The work started off and was primarily driven in terms of clinical recruitment by Jenny Hudson in Sydney and by Kathy Creswell in Reading. And we recruited around 2,000 children who are being treated with a manualised form of CBT for a child anxiety disorder. And the groups of treatment fell into 3 forms of delivery, or modes of delivery. There was the guided self-help, which is the mum working through a book, for example, Kathy Creswell's Overcoming, with some remote therapist assistance. There was group treatment, which is quite common, particularly in Australia, and the guided remote was very common in Australia as well, where often the physical distances simply mean that that's the only realistic approach to treatment. And then, of course, there was a high level of individual treatment. And the extent to which these 3 types of treatment were offered across the different sites varied quite considerably. So we controlled for site and what was on offer and pre-treatment characteristics of the children in all of our analyses. And I'm just going to show you the genetic analysis because that was what really gave us our first hint that this is a useful approach. And because I think some of you might not be familiar with this type of data, I'll walk you through it. So here we have the whole genome, which is 23 chromosomes, 1 through to 22, and then the X chromosome is here. And this is the log of the p-value. So the p-value you would usually see in experimental medicine would be 0.05, and that would be because you're assuming that it's okay to have one test be wrong out of 20. That's what your 0.05 means. You're prepared to risk being wrong 1 time out of 20. I'm sure you're all familiar with the concept of Bonferroni correction, that if you do 100 tests, instead of having a p-value of 0.05, you would divide it by 100, and so you'd have a more stringent p-value. Well, in a genome-wide association analysis, you do 1 million tests. So 0.05 divided by a million is 5 times 10 to the minus 8. And so that's why this is the log of the p-value. So these individual data points relate to the statistical significance corrected for having done a million tests. And so this is the genome-wide significance level. This means that an individual variant is statistically significantly accounting for variance on its own. Now, this data set was around 1,000 individuals in the end, although we had data from 2,000 by the time we had the outcome data and clean genomic data. We lost quite a lot of information, and we now know that we need far more people than this. But what we see here is a very typical pattern. You see some areas where these individual genetic variants are coming up above the rest of the information. There's a little bit of true information coming up above all this noise down here. These variants here are not useful in understanding treatment response, but these ones nearer the top, more of them are useful than not. There will be some false positives in there, but there'll be a lot more true information in there as well. And what we've discovered in the latest 5 years or so of genotyping work of this kind is that you really need to be looking at samples of 10 or more thousand patients to be having statistical power to look at a single variant and have it come above this magic genome-wide significant line. But what you can do even in slightly smaller samples is to combine the information. So you can combine the more useful end of the distribution of information. So that's what we did in this study. And we combined information from a different data set that provided an insight into something called differential susceptibility that we thought would be relevant to treatment response. Now, differential susceptibility refers to the fact that individuals may not just be vulnerable to poor environments in the way that the diathesis stress model would propose, but they may be responsive to environments overall. And so the idea is that individuals who are very responsive will do really well when they're looked after carefully and they're in positive enriching environments in the way that an orchid would. Whereas if one sits on my windowsill, it unfortunately ends up looking like this. And so individuals who are very responsive but are in less enriching environments or in negative environments, there's likely to be a poorer outcome. And these individuals can be contrasted against individuals who are quite resilient regardless of the environment. So it won't make that much difference if they're in a good environment, but it also won't affect them that badly if they're in a poor environment. And so you have a distribution of how responsive individuals are to the environment. And there was a lot of thinking around this idea about 5-6 years ago when we were also thinking about what genes to look at with regard to treatment response. And there was also a lot of theorising about the idea that predictors of response to psychological treatment might be reflecting the predictors that it led people to those disorders in the first place. So the idea that cause might inform cure. And this theory is a quite nice way of encapsulating that idea. So what we've got here is data from our treatment response study, which I'll walk you through. At this end of the an analysis that looked at genetic variants associated with responsivity to the environment. So according to their genetic susceptibility score for this measure, they should be very unresponsive to the environment. In contrast, these individuals are highly responsive to their environment, so they might be more at risk if they're in negative environments. And when we looked at this genetic score, we found that these individuals, the very responsive individuals, have particularly poor emotional outcomes in the presence of negative parenting. But this figure that I'm showing you here relates to the treatment groups that I went over before. So we have our individual CBT, our group CBT, and our remote delivery. And you can see for the unresponsive group, it really doesn't make any difference which type of treatment they receive. This is change in their anxiety symptom scores and the clinical severity of their presentation from the ADIS, and you can see that they're all around 3 to 3.5, and this is a scale where 4 is where diagnoses are made, up to 8 and 0 is none. So a drop of 3, 3.5 is good. Most of these children are doing pretty well. But what's even more striking is that in the very responsive children, from the point of view of this genetic score that we created, those that received individual CBT did really, really well. They had huge reductions in their symptoms. And our interpretation of that is that these children are more vulnerable and therefore need the highest quality possible treatment, and we know that mothers of anxious children are often anxious themselves, making treatment delivery quite difficult for them. So where are we going with this work? We need larger samples. It's now clear that we need tens of thousands of samples to do this kind of genetic work in a well-powered way. So we have just built an online resource, an online platform to recruit through IAPT. We're starting within SLAM. This is in the adult service at present, but we have plans to move into recruitment of children as well. Individuals will be able to come onto the website, sign up, complete some questionnaires. They provide consent for us to access their medical records and to recontact them for future research. And they provide a saliva sample. And we can use all of this information to try and predict who benefits from which arms and which forms of treatment within the IAPT service. And this could lead, I hope in time, to a situation where when a child comes into a clinician and you need to make a decision about which form of treatment to provide. At present, there's not a lot of evidence to guide you, but in time we could have an algorithm that combines demographic, clinical and also genetic data to give some sense of likelihood of good outcome from each of these types of treatment, so that you can make a more informed decision about who needs which treatment at which point. So thank you for your time.